Hi friends, welcome to Biology Tutor. If you like this channel, please share and subscribe. Don't forget to click the bell button for notifications. Today we are going to discuss about staining. Before going to the staining techniques, we have to fix the specimens. First step is the fixation process. Fixation means it is the process by which the internal and external structures of specimens are preserved and fixed in position. Why should we fix the specimens? It helps to inactivate enzymes that disrupts cell morphology and also helps to toughen the cell structures. For that purpose, we have to fix the specimens before observation through microscopes. It helps to retain the stain easily and accurately at desired position of the specimen. There are two different methods are there. The first one is heat fixation and the second one is known as chemical fixation. What is heat fixation? How to perform the heat fixation process? Heat fixation means it is used to observe bacteria and archaebacteria. Here smear is gently heated. It helps to inactivate enzymes and preserves morphology. It destroys proteins in subcellular structures, thus distort the appearance. That is the peculiarity of heat fixation. Heat fixation is employed in the case of bacteria and archaebacteria. Next step is chemical fixation. What is chemical fixation? It is often used to observe large and delicate microorganisms like protists. Chemical fixation is used in the case of protists. Heat fixation is normally used in the case of bacteria and archaebacteria. Archaebacteria means ancient bacteria. Chemical preservatives penetrate cells and inactivate proteins and lipids inside the cell. Examples are ethanol. Chemical fixatives examples are ethanol, acetic acid, mercuric chloride, formaldehyde and glutaraldehyde. These chemicals are used as chemical fixatives. Dyes used. We need dyes to observe microorganisms on other cell bodies using microscopes. Dyes should have two features in common. Dyes have chromophore groups. It imparts coloration. First feature is these are chemical moieties with conjugated double bonds that give the dye its color. And second feature is they bind cells by ionic, covalent or hydrophobic bonds. According to the ionizable properties, the dyes are classified into two. A. Basic dyes. Example, methylene blue, basic fuchsin, crystal violet, saffronin, malachite green. It is very important. Characteristics. Basic dyes have positively charged groups, binds to negatively charged molecules such as nucleic acids, proteins and surface of bacterial and archaebacterial cells. That is a peculiarity of basic dyes. It has positively charged. Which among the following is positively charged dyes? Basic dyes. Basic dyes are positively charged and examples are methylene blue, basic fuchsin, crystal violet, saffronin and malachite green. Malachite green is used in spore staining. Ionizable dyes. Acidic dyes. Example, eosin, rose bengal and acidic fuchsin possess groups such as carboxyl and phenolic hydroxyls. Characteristics. In ionized form, they have negatively charged and bind to positively charged cell structures. Acidic dyes are negatively charged ones and examples are eosin, rose bengal, acid fusion. And one more types of dyes are there, that are known as neutral dyes. Example, Giemsa stain, eosinate of methylene blue, carries both positive and negative charges. Neutral dyes, the peculiarity is it contains positive and negative charges. Stains, usually it stains nucleic acids and cytoplasm. It stains nucleic acids and cytoplasm. Giemsa stain, discovered by Gustav Giemsa. It is very important, used for demonstration of malarial parasites in blood. It is widely used for the routine examination of blood smear. Types of staining techniques. There are two methods, simple staining and differential staining are there. First one, simple staining. We are going to discuss about simple staining today. Here only single stain is used. It is used for visualization of morphological shape and arrangement of cells or bacteria. Simple staining. A single dye is used and cells are stained. Fixed smear is covered with stain for a short time and excess stain is washed off with water. Here slide is blotted dry and basic dyes such as crystal violet, methylene blue and cardboard fuchsin are frequently used. In right side you can see the simple stained slide. This is a picture of simple staining. Negative staining. A single dye is used. This is also a simple staining technique and only one dye is used. A single dye is used, but here unstained cells appear as bright object against a dark background. Here background is dark and the cells appear bright in color. Here Negrosin and Indian ink are used. What is electron cryotomography? Advances in electron microscopy leads to the development of electron cryotomography. Here samples are prepared by dipping into an extremely cold liquid. Normally ethane is used. Sample is kept frozen while being examined in electron cryotomography. Vitreous ice keeps the native structure of the specimen. Why should we cool these specimens? It will help to keep the native structure of the specimen and also helps to immobilize the particular specimens. So we can easily observe using electron cryotomography. Cryo means cold. It can be viewed in the high vacuum of the electron microscope. Here object is viewed from many directions. Individual images are processed and merged using computer programs. Finally, we obtain merged 3D image of the specimen. Ultrastructure of bacterial and archaeal cell can easily be viewed using this technique. This is a 3D electron tomogram of a neuron-like culture cell determined by cryo-electron tomography. Scanning probe microscopy. This type of microscopes are powerful microscopes. These microscopes can measure surface features of an object by moving a sharp probe over the object's surface. Here the peculiarity is a probe is there. That's why it is called as scanning probe microscopy. That probe or projections which helps to observe particular specimens or cells. Example for this type of microscope is scanning tunneling microscope. Scanning tunneling microscope. Magnification is 100 million times. This can be used to observe even atoms of small size. Scanning tunneling microscope. This type of microscopes have a needle-like probe with a point and can observe a single atom at a time. That is a peculiarity of scanning tunneling microscope. This was invented by Gerd Binning and Heinrich Rohrer. Gerd Binning and Heinrich Rohrer invented scanning tunneling microscope. Atomic force microscope. Atomic force microscope is a second type of scanning probe microscope or SPM, which moves a sharp probe. Here probe is there, which moves a sharp probe over the specimen. AFM is used to study, AF means atomic force microscope, is used to study the interactions of proteins, behavior of living bacteria, and also used to visualize membrane proteins such as aquaporins. This is the diagram of atomic force microscope. You can see the tip that probes. We can easily visualize atoms. Summary of all the microscopes. You have to be very careful and you have to learn this summary before going to exam. This diagram describes the properties of light microscope, like magnification of objective eyepiece, total magnification, numerical aperture, mirror used, focal length, working distance, iris diaphragm, position of condenser and maximum resolution, etc. Just go through this before going to exam. It is very, very important. Next, the bright field, dark field, phase contrast, fluorescence, term and sum. Here you have to learn the magnification and resolving power. Next, the property of STM, scanning tunneling microscopy, atomic force microscopy, transmission electron microscopy and scanning electron microscopy. You have to remember some points from these chart like resolution and dimension. Thank you. If you like this channel, please share and subscribe. Don't forget to click the bell button for notifications.